Myself, I'm Laurel Richardson. I'm the Director of Community Outreach for the Charcot-Marie-Tooth Association. And I thank you for being with us today. I'm going to turn it over to my colleague, Gina Sweeney.
Hello, everyone. As a patient myself and as a mother of an 18-year-old daughter living with CMT 1A, I welcome you all to the CMTA 1A Town Hall meeting. As Laurel said, my name is Gina Sweeney, and I am the Chief Engagement and Gift Officer for the CMTA. So I'm just going to start with just a little story because I only have a minute, but a little over 20 years ago, I made the choice to raise my hand to be a branch leader for the CMTA, and it was truly one of the best choices I made for myself because as someone living with a spontaneous case of CMT, not only did I find my community and my people, but I was involved to see STAR, Strategy to Accelerate Research, born. And that was in 2018. And learning about STAR, my first thought was, well, the doctors can't help me help my daughter, Riley. She was 3 at the time. They can't give her medicine to treat it or to cure it, but I know that the CMTA can. And I began my journey by jumping in with 2 feet, creating more awareness for CMT everywhere and anywhere I could, personally donating and fundraising for the CMTA. And here I am now working for the CMTA. Never saw that one coming, but it's more than a job just for me and for the staff and for the board. It's my cause, it's our cause, it's our family's cause, it's your cause. So today you'll hear why we are all in, and you'll hear from some of the people I respect the most, the best of the best, the CMTA leaders and the researchers, all solely working for you and your families. And if you don't know the CMTA or not fully connected, I encourage each of you to get to know us. Because as we are researching for treatments tomorrow, we are all here for you today, whether it's community connection, education, resources, or a friendly voice on the other end of the line. We are here and we are family. Before I turn it over to Dr. Catherine Forsay, Chief Research Officer for the CMTA, a committed community member sent me these cute little hope cards because she sees the CMTA as hope. My job is to pay it forward so that I can create hope for you. So I'm going to read that. After the final no, there comes a yes. And on that yes, the future world depends. So with that, take it away, Dr. Forsay.
Thank you, Gina. I always like it when you call me Dr. Forsay. It always makes me smile. Thank you. So it's really great to be here with you all today. As Gina said, I'm Catherine Forsay. I'm the CMTA's Chief Research Officer. I'm also a CMT 1A patient and parent myself, so I'm really happy to be spending time with you all today, my fellow 1A people. It's great to see that so many people have joined us live, and I know that there's lots of people who couldn't join us live who signed up to receive a copy of this recording. So it's my absolute honour to be able to share our programme for the day and introduce our speakers. We're really grateful for their time and their tireless work to advance CMT research, therapy development, and clinical trial readiness for CMT 1A. I will shortly be handing you over to David Herman, MD, from the University of Rochester and a member of the CMTA's Clinical Expert Board. David will be introducing CMT 1A, its cause, symptoms, burden of illness, and some of the current care options that there are. This will lead into the main CMT 1A research update, which has kindly been provided by John Sfarren from the University of Wisconsin, and John's also chair of the CMTA's Scientific Advisory Board. We'll then hop back to David for a short piece on the ACT-CMT study and clinical trial readiness before we hear from Nikolay Zelensky, who's from Argenx, a Swiss-based biotech firm who are working to develop a new treatment for 1A. And then we'll hear from Gilles Bouchard. Gilles' chairman of the CMTA's board of directors, and he'll provide that broader picture view of company interest in CMT 1A. Finally, you'll get me back, and I'm going to share a little bit about patients as partners in research, which is one of the CMTA's important programs, and how you can get involved in research for CMT 1A. We're then going to move to a short Q&A section, and we'll be asking you to pop your questions in the chat, and I will put those questions to the panel. After the Q&A, we'll have a very special community announcement from Gina Sweeney before we close the session. So without further ado, I'd like to hand you over to David Herman, who's going to provide that introduction to CMT 1A. Over to you, David.
Thank you, Catherine. It's a pleasure to be here today. By way of background, CMT1A is the most common form of CMT and accounts for about 40% of all CMT. It's typically inherited in an autosomal dominant fashion, although there can be individual or new, what we call de novo cases without a known family history. It is due to a 1.4 megabase duplication within the peripheral myelin 22 or PMP22 gene on chromosome 17. And this leads to an overexpression of PMP22, therefore too much PMP22. And PMP22 overexpression in rodent models and in mice has been shown to cause a demyelinating neuropathy that is similar to CMT1A in humans. And strategies to reduce the amount of PMP22 expression has been shown to improve or correct multiple animal models of CMT1A. And therefore, the main strategy to try and treat CMT1A in humans has been directed at trying to decrease the overexpression of PMP22. But there are many other complementary strategies that are being pursued. Importantly, when we think about treating CMT1A, we know that disability relates to the degree of axon loss. And therefore, as we think about interventions for CMT1A, these should be aimed to either prevent, reverse, or slow down the loss of axons. CMT1A has multiple impacts for patients, and we have studied the patient's perspective on CMT1A. In one study, we found that quality of life is substantially impaired by CMT1A. Major concerns are difficulties with gait and balance, foot and ankle weakness, inability to do activities, and hand and finger weakness, among a multitude of challenges that patients with CMT1A experience. Beyond impacts for individuals and families with CMT1A, there is a broader societal impact, and this has been recently studied in Germany in a systematic fashion. And what that study showed in Germany, which we believe applies to the United States and other parts of the world, is that CMT neuropathies are associated with substantial health economic burden for patients and society as a whole. Therefore, emphasizing the broader importance of developing effective strategies to treat and manage CMT1A. Currently, we lack a disease-modifying treatment for CMT1A, and that is, we'll learn a lot about some of the advances towards treatment for CMT1A today and all the ongoing work. But patients with CMT1A, in my experience, can benefit substantially from a multidisciplinary care environment. We at University of Rochester, but at multiple other centers, have such a multidisciplinary care environment, and it really depends on a team of specialists to optimize function of individuals affected by CMT1A and to try and target symptoms that are impairing quality of life. I'll now turn over the discussion to my colleague, Dr. John Sfarron, who will discuss all the exciting advances towards therapy for CMT1A.
Thank you. So I'm really privileged to present the work that we've done on the CMTA side, basically to really attack CMT1A on really multiple fronts. And what I want to cover today is multiple initiatives that have involved genetic therapies for CMT1A. This is coupled with company partnerships that are an increasingly important work of our organization. One thing that is critical to help these companies is to really develop biomarkers and other tools for clinical trials to make those better and more efficient and more optimized. And also there's still significant initiatives for drug development as well. So on the next slide, please. So one of the things that really prompted a lot of excitement in this field is a development of gene therapies for a disease called spinal muscular atrophy. SMA is a disease that actually affects the same motor neurons that come from the spinal cord out to your muscles. And SMA is really a devastating disease that typically affects children. And one of the things that's been really spectacular is the development of novel therapies for this disease. And these have become approved by companies, and a number of patients with that have really benefit from this. And a couple terms I want to introduce to you is AAV gene replacement therapy. This was developed by Avexis, and also ASO therapy. These are genetic therapies that allow treatment of this kind of disease. Also, this type of therapy is also being employed for actually a type of CMT known as giant axonal neuropathy, and there have been clinical trials ongoing for that. Now, we wanted to take these advances in these other diseases and really apply those to many types of CMT, including CMT1A. And I hope to give you just a brief overview of the efforts that have been ongoing in this endeavor. Next slide, please. So ASO stands for antisense oligonucleotides. Another version of this technology is known as RNA interference. And essentially, these are ways where you can actually target a gene that's causing the problem. As Dr. Herman had mentioned, the excess of PMP22 is what causes CMT1A. And you can target a gene like PMP22 by using these types of genetic therapies. And in fact, we were able to collaborate as an organization with Ionis that provided really the first proof that in a rodent model of CMT1A, actually 2 rodent models of CMT1A, we could really get dramatic effects, dramatic improvement in nerve function as a function of targeting PMP22 in this manner. So this was a very exciting finding that was published several years ago now. And this has really developed into a full-blown initiative that's taken apart not only of our organization, but also multiple companies that have joined in. Next slide, please. So one of the things I just wanted to illustrate that the path that has emerged from these studies really is that initially that we were following this opportunity that was presented by SMA therapies. And then we worked with Ionis, to develop those in these preclinical treatments of CMT1A. Now, this immediately provided a couple of challenges that it took us some time to overcome. One of those is that we had a challenge in terms of really efficiently delivering these therapeutic agents to nerve. Getting into the brain or getting into the nerve is a challenge for different kinds of drugs and this type of therapy as well. The other thing that was a challenge at the time is that we really did not have adequate clinical assessments to be able to assess progress in a clinical trial. And so these were 2 major impediments to bring that kind of therapy into clinical trials. But what happened is with the publication of this study back in 2018, we saw really in the last several years now, really an explosion of interest on the part of many companies that work in this space, ASOs and RNAi. And some of them have different strategies for improving delivery of RNAi to nerve. And I've listed a few companies on this diagram here. X, Y, and Z indicates we know of many other companies that are working in this area, and we can't disclose their names at this time. But probably the company that's become the most advanced in this is DTX Pharma. And they developed a RNAi therapy that showed promise in their work. And in fact, they were planning to bring this to clinical trials even as early as the coming year. And in the meantime, this was company was now acquired by Novartis, which is a major pharma firm, international firm, and which has really the resources to put their shoulders, behind this effort to really push this into clinical trials. In the meantime, in parallel, the work of a lot of people led us to progress in clinical assessments, and I'll mention a little bit about MRI and other assays that we think are really going to make clinical trials for CMT1A much more effective. So we've always been identifying these challenges and moving forward to try to resolve those. I do want to feature Novartis a little bit, partly because the global head of neuroscience for that organization and who spearheaded this acquisition is Dr. Robert Balow. He's very well known to us in the field. He was a neurologist who had a clinic in CMT and ALS, initially at Washington University in St. Louis, and then later in Cedars-Sinai in California. And he became global head of Novartis. He was actually a former member of our scientific advisory board. So for those of us in the field, we're extremely excited that he was involved in really spearheading this acquisition. And we look forward to working with Novartis as they hopefully develop their clinical trial designs. And we're actively in discussion with that. Although this is still a little bit early days since the acquisition, so the plans have yet to be formed. Next slide, please. So aside from this RNAi initiative as well, I did want to highlight that we also have been supporting other kinds of approaches. One is a sort of a viral gene therapy approach, which is AAV. We've been fortunate to support a neurologist in Cyprus, Dr. Cleopa, and he has collaborated with Scott Harper at Nationwide Children's to develop a viral gene therapy that accomplishes the same goal. And in fact, this approach is being developed by Armatus, a company that's moving forward to try to bring this to clinical trials. So this is a parallel approach to the one that I just mentioned before. And in fact, the other technology that many of us have heard about, which is CRISPR-Cas9 or gene editing, this has really been a revolutionary advance, which is just starting to be applied in medicine. And we've been privileged to work with a company called Toolgen, which has actually published some of their work. They presented recently at the Peripheral Nerve Society meeting, and they are actually, working with us to try to bring this technology to CMT1A. All of these things still have some challenges. One of those is to be able to deliver these therapeutic agents to Schwann cells in the nerve. And all of these approaches have a similar challenge in that respect. And we've also been investing in novel technologies for this. We've been recently supported a project using nanoparticles as a way of sort of delivering these therapeutic agents to the nerve. And this is a joint project that's been supported by the CMTA in collaboration with the Muscular Dystrophy Association. Next slide, please. The other critical point that I've alluded to before is we really needed to have better assessments of how we measure neuropathy and know if actually during a clinical trial, if people are responding to that treatment. This is critical not only for patients, but also for the companies that are investing their money in these clinical trials. They need to have a way to measure success. And there's been a series of efforts by neurologists all over the world. For example, development of neuropathy scores. This has been expanded to pediatric and infant assessments as well. But this still has remained a challenge because CMT is known as a slowly progressive disease. And we needed additional measures to be able to see if we can measure improvement. And really, this is a critical aspect. So if we don't have a good measures for clinical trials, companies are not going to invest in CMT1A. And it was really crucial for us to invest in this as well. And the CMTA has heavily invested in this area. I just want to give you a couple examples of that in the next couple slides. So one of those measures on the right side of the screen is using MRI of calf muscle. It turns out you can measure the amount of muscle in the calf using MRI very sensitively. And when there's not enough muscle there, that gets replaced by adipose tissue. And this has turned out to be a very sensitive measure of progression. This was developed by Dr. Riley at University College London, and then has been applied in other centers across the US and elsewhere. And this has been an exciting development. In fact, this measure is being used for clinical trials and some types of neuropathy, and we're hoping this will be incorporated into CMT1A clinical trials. The other thing that has happened in collaboration with many of these neurologists that I mentioned is development of new things, not only neuropathy scores, but Dr. Herman and Katie Einkinger at University of Rochester have been developing ways of using wearable sensors to measure things like activity or balance or those sorts of things, to be able to, again, develop more quantitative data that can be used to power and enable clinical trials. Next slide. So the other things that we've been looking at is looking at things that we can find in blood samples or in skin samples. Some of you may have gone to centers where you've been asked to be given a blood sample to be used for biomarker studies. And we've been able to develop ways to assess how axons are doing. Dr. Herman mentioned that axon loss is a critical aspect of the progression of CMT. And we've looked at other biomarkers that are sort of that come up in CMT1A. And what we hope will happen in a successful clinical trials is that these biomarkers will respond to that and give us really an early readout of whether the therapy is actually working for patients. And again, a combination of these things are really going to be required to help us to design the most critical aspects of clinical trial design. Next slide. So this is my imagination of what a successful clinical trial will look like in CMT1A. If we look at the ASO aspect of this, the first thing that we can do is reduce the levels of PP22. This will improve the myelin that's formed around nerves. This will ideally lead to improved nerve function and also the preservation of those axons. And then hopefully patients will begin to notice. They'll actually notice that they hopefully will have a better function, or at least the progression of the CMT will be arrested. And having different measures that we can assess along this trajectory will be really critical for companies to be able to know both at early stages and later stages of clinical trial whether their therapy is actually working. And that's one of the things that we've been really looking at, is creating an array of different assessments that we can use using protein and transcript measurements in order to enable that. Next slide. So I did want to just say that not only are we working on the genetic front, we are also doing things like drug testing with pharma partners. This has been a really active area. Our board chair, Gilles Bouchard, will say more about that later on. There have been some advances in HDAC6 inhibitors. The recent meeting of the Peripheral Nerve Society, there were several companies that presented some work in that area. One of these compounds is actually in phase 1 safety trials. We have a project in our lab to try to find small molecules to reduce PMP22. Another project that's being supported initially at the University of Buffalo in the Feltri laboratory has really shown some promise with some drugs that are actually approved. And we're actively working with companies, again, that work in the ASO RNAi space. So I can't convey all the initiatives that we have in the brief time we have together, but I just wanted to say that we're really relying on not just one kind of therapy. We're very testing and trying to support initiatives across the board. And hopefully we can race to see who can get to clinical trials and show improvement fastest. Next slide, please. So again, I just want to reiterate that, we really have a multi-front effort incorporating all of these initiatives and partnerships. And it's really an exciting time for us. Many of these efforts are finally coming to fruition. We are very eager for this to move as quickly as possible. But we have to have had to recognize the challenges along the way and address those. Next slide, please. I'm going to hand this over back to Dr. Herman. He and Michael Shai have been really leaders in the clinical effort. And so I'd like to ask him if he could just summarize sort of the global effort that we've had to develop clinical trial readiness.
Thank you, John. So in order to accomplish much of what John has outlined here and to be ready for clinical trials, we needed to establish the infrastructure to do this. And so about 14 years ago, under the leadership of my colleague, Michael Shai at the University of Rochester, but with centers such as ours and others, we formed what is known as the Inherited Neuropathies Consortium. This has been supported via the NIH through their Rare Disease Clinical Research Network program. And there are over 20 centers globally that collaborate to do studies to plan for and prepare for clinical trials in CMT1A and in fact other forms of CMT. Next slide, please. So one major initiative I'd like to highlight focused on CMT1A is a study known as Accelerate Clinical Trials in Charcot-Marie-Tooth Disease, or ACT-CMT. This is a study that we lead out at University of Rochester, but we have 5 other collaborating centers within the Inherited Neuropathies Consortium who play critical roles in this study. And this study has aimed to really understand the natural history and course of CMT1A, and also to validate various of the assessments that Dr. Sfarin highlighted in order to be ready for clinical trials. So notwithstanding the COVID-19 pandemic, this study started in late 2019 and is now actually in its final year. We enrolled 215 individuals, adults with CMT1A, and we are developing very detailed information about how our different clinical assessments, patient-reported assessments, functional outcome measures such as our CMT functional outcome measure, and indeed our biomarkers, MRI of the lower extremity muscles, and a technique called in vivo confocal reflectance microscopy of Meissner corpuscles. These are sensory nerve endings in the skin, function to assess the course of CMT1A. We hope that from the information that's emerging, that we will be able to design and plan clinical trials that will be able to detect whether treatments are working in CMT1A. And much of this information that is now being generated is being used in collaboration with industry partners to help plan for hopefully multiple upcoming trials in CMT1A.
Thank you very much, David. That was really useful, and John, for your perspectives. I'm now going to hand over to Nikolai Zakowski from Argenix, who's going to give us an overview of one of the biopharma partners that we're working with to attack CMT1A. Nikolai.
Hello, everyone. It is my pleasure to be part of today's event. And thank you very much, Dr. Forsyth, for the invitation to present our research to your community. Next slide, please. So today I would like to introduce you to a molecule called alpha-1 antitrypsin, also commonly known as AAT, and its role in regulating inflammation. So by now you will all appreciate the central role that PMP22 overexpression plays in propagating CMT1A's pathology. Both Dr. Herman and Dr. Sfarin both highlighted the importance of regulating PMP22 and any potential treatment that reduces the amount of PMP22 to be effective. But here we took a little bit of a different approach, looking downstream from PMP22. In other words, trying to mitigate the effect of PMP22 overexpression rather than tackling PMP22 itself. So back in 2019, a pivotal study led by researchers from Johns Hopkins, published in the prestigious Nature Biomedical Engineering journal, investigated what PMP22 overexpression actually does. So specifically to Schwann cells, what factors in essence are affected as a result? And the conclusion was quite fascinating. It revealed that in fact, PMP22 overexpression leads to a massive upregulation of pro-inflammatory cytokines. In effect, it primarily initiates a cascade of local inflammation. And coming back to AAT, and to put it in more relatable terms, I would like to use a simple analogy, as you see here in the image. Just one slide before, thanks. One can really view PMP22 overexpression as the hot and scorching desert sun. So it is far from kind to any attempts of natural growth. Nevertheless, if one is to water, say, in this example, an apple tree on a regular basis, it gives us the chance to thrive in these difficult desert conditions. Next slide, please. So we did our research in vitro, so meaning on cell cultures, and we found that AT is particularly relevant for CMT1A, as it has a unique dual specificity for both Schwann cells and macrophages. First, we have shown that AT has the ability to act directly on Schwann cells, blocking the feed-forward loop propagated by pro-inflammatory cytokines that are upregulated as a result of PMP22's overexpression. Second, and importantly, we have shown that AT has the ability to act directly on macrophages, switching them from their M1 attacking state to their M2 regenerative state. And macrophages, just to highlight here, are especially important in CMT1A, as they are the cells that are presumed to cause the most damage to the axons while in their M1 state. So by regulating macrophages, AT is not only able to prevent damage to axons, but it's also able to restore their regeneration. Next slide, please. And this dual therapeutic action that I described in my previous slide of AT was actually able to not only slow down the progression of the disease, but to actually reverse it in a mouse model of CMT1A. And today, in the interest of time, I would like to share with you just a few key observations after 2 weeks of AAT treatment. So the axons, the myelin, the muscles of CMT1A mouse showed an improvement by 50%. So that's halfway back to healthy. We have a lot more compelling data that shows recovery in other important parameters, including different pro-inflammatory cytokines such as interleukin-6, TNF-alpha, or the biomarker NFL that you've heard about before. But next slide, please. So we shared our full data package with the FDA, and in July have been granted the orphan drug designation for AAT. And this is a very important step for us as we prepare to further develop AAT for the treatment of CMT1A patients. Next slide, please. And last but not least, a few words about our company, Agironics, which is based in Geneva, Switzerland. We work in close collaboration with the Geneva University Hospital, which is the largest in Switzerland in the country and one of the largest hospitals in Europe. And I would like to really take this opportunity to thank my team here in Geneva. It has been really an exciting journey thus far, from the first idea to really tap into this natural ability of the body to regenerate the peripheral nervous system, which came back in 2020, to really taking a deeper dive and studying the underlying molecular mechanisms that make AAT a uniquely promising treatment for CMT1A. So many thanks again to the CMT Association for allowing me to share a story with you today. This concludes my part of the presentation, but I will be happy to take any follow-up questions during the Q&A. Thank you.
Thank you, Nikolai, and thank you everyone who's going to be staying for the Q&A at the end. We really appreciate your and your company's work in 1A and bringing this drug to our patient population, and wish you every success as your research continues. We're going to move now to Gilles Bouchard, who is chairman of the CMTA Board of Directors. Gilles.
Thank you, Catherine. Let me share my screen. Here we go. Thank you. So I want to talk about momentum and partners. And I only have 2 slides, so I will spend a bit more time on each slide. The first point I want to make, and if you recall, when we started STAR more than 10 years ago, the whole idea behind STAR is how do we make CMT attractive to partners? We cannot do it alone, and I will explain why. Why are partners so important? Let's go back to this a little bit. The very first reason we started this is, and this has been studied very extensively, and the numbers are a little bit over the map, but it takes an average over 10 years and $1 billion to bring a drug to market. And that's if it's successful. And unfortunately, about 90% of drugs fail in clinical trials. So as you can imagine, and you can see, this is a very expensive, very lengthy, very risky business. And we cannot do it alone. We need to bring partners to this who are willing to help share the load and carry this with us. What we've also found is, as you get closer to the end goal, it takes very unique capabilities to bring the large-scale clinical trials and also the large drug distribution to market. Even actually, some medium-sized drug companies do partners with larger companies to bring drugs to market. So partners are required all along the value chain here. Now, the third point, which we've learned a lot, and the doctors talk about this a little bit, we will most likely need a portfolio of technologies and treatments to help people. First, because we cannot put all eggs in one basket. Every single opportunity I will discuss is exciting, but it's also very likely not to work. And the main issue, by the way, when things don't work is there's a lot of drugs that can mitigate PMP22. We know many of them, but yet we haven't found a way to deliver them safely in the right doses to patients. So delivery is critical. And of course, clinical trial development is critical. So there's a lot of steps left to be done here. And because this is so risky, we need to diversify this risk. It's a little bit like when you plan your retirement. You don't want to invest all your money in a very high risky stock. You'll probably have a very painful retirement. It's the same thing with drug development. You want to invest in many, many different areas so that you can mitigate the risk across many, many different approaches. And that's what we're doing right now. But also we know that most likely we're not going to come up with one magic pill or injection and the next day CMT1A will disappear for all the patients. Unfortunately, I think we'll need many, many different approaches, many different technologies that will be customized to everybody over time. So there will be many different partners, many different drugs, many different clinical trials as a solution. And of course, we need to address all kinds of CMT. Even though it's a CMT1A patient, sometimes we will benefit from approaches that were first initialized in other CMTs. And we see that more and more. I'll give you some examples. And the fourth point that's interesting is every time we've had a major partnership, even though they have not necessarily led to a drug reaching the market, they've made us make giant steps forward for the ecosystem. And I'll give you 2 examples. You remember a few years ago, we had a couple partnerships. One was with Sanofi Genzyme, and that partnership helped us tremendously define the testing infrastructure that I'll describe later, but also all the work that David mentioned with the INC and clinical development was really informed tremendously by the work with such a professional organization, and is helping us even today. Same thing as John mentioned, the work we did with Ionis a few years ago, which really proved the concept that RNA technology would work in CMT1A, and now it's really led to this explosion of companies that I'll share with you that are working on this. Okay, so this is why partners are important. Now, how do we make CMT attractive to partners? That's the next question. Okay. So we came up with 5 different areas that companies need help, and they're all in there. Starting with the top and going clockwise, usually companies first need advice. They tend to know their own technology, but they don't know CMT. So that's why we assembled such an amazing panel of experts in our scientific advisory board. You got 2 great examples here online, but we have over 30 people that can help companies understand our ecosystem better. And then especially for small companies, often they need money and fundraising, and we're providing more and more of that. Or big companies more and more come to us and say, what do you have in your portfolio that I can potentially license or take to market? And then the green one is very important, is this preclinical testing infrastructure, which we developed over the years, where companies can come in and work with us and very quickly, very inexpensively test their drugs on CMT. And in every given time, you'll see a bit later, there's probably half a dozen companies who are working and testing CMT drugs with us, thanks to all the work we developed and invested in STAR over the last 12 years. And then the next step is clinical trials. You heard a lot from John and from David on this. This is the last frontier. There hasn't been a successful clinical trial in CMT yet. It is really something that is still an inhibitor for company to work on CMT. That's why we invested so much in what John talked about, biomarkers, so that we can have more reliable, faster, less expensive clinical trials. And we've had tremendous progress on this, thanks again to major investment from STAR on this. And also we've been supportive from the beginning from the ANC together with the NIH and the MDA, and they've created a wonderful infrastructure that is now able to host clinical trials for companies, with a couple going on already. And last but not least, it's you guys, access to the community. I can tell you how much companies want to talk to you, want to learn from you, and luckily there's more and more of this. So you will have a growing role to play thanks to this, and we see a lot of them now. So that's how we help. And the next question, so is it working? And the answer is yes. And it's working a lot better than it did 10 years ago when we had nothing to show. So I'm going to show you some examples of companies, what they do, and I will help them. So there are three areas of technologies that you've heard from John, the ASO-RNA, the gene therapy, and the small molecules. So let me kind of map all this for you. So on the RNA side, there are 6 companies we're working with, and I'm only listing the company, but also that's why I call it code on the left side. You can see what we do with them, because I want to be very clear. For us, when we say partner, it doesn't mean we just put their logo on our website. Okay, we actually roll up our sleeves, work with them, add value, and try to help them move forward. So it's really working together closely with those companies, and you can see through the color coding what kind of the resources that you've helped us develop over the years they use from the CMTA from STAR. So actually, John had the same list. On the RNA side, you got DTX Novartis, which is a very exciting development. You got a couple of small companies, Shift and Mastrina out of France. And then you got 3 major companies who are, as you can see, already testing those kind of technology with us. And they don't want us to publish their name. In general, what you'll find is smaller companies want to go public fairly quickly because they want to raise money, but larger pharmas want to wait because first they want to make sure it works, but also they want to protect the IP. So in general, the companies that are not named in a good way are very big, serious, big pharmas. You'll see 6 of them on our chart today. And I looked this morning, the market cap of those 6 companies is over $300 billion. So we're talking about really serious players now working with us on CMT1A. It's incredible. Okay. So this is the RNA picture. Gene therapy, again, John mentioned many of that. Armatus, we're helping with the community. We're also supporting them financially. Toolgen, they've been doing a lot of testing with us, still going on, using expertise. Gladstone's a great example. Gladstone is not a company, but it's a leading lab in CRISPR. For example, Jennifer Doudna, you heard about, is part of Gladstone. So we're talking about the best of the best in CRISPR. And they came to us and said, we looked at the whole space of diseases. We think CMT is a great target for CRISPR, and we want to work with you and all the tools you've developed. So we've had this great relationship with Gladstone, starting CMT2s and now moving to CMT1s on CRISPR. And then there's a new company called NervoSave, which is actually doing injections in the nerves. So a very different approach. And again, always interesting to look at people who look at kind of different things, because it provides different approaches for potentially treatments. So that's the gene therapy space. And then in small molecules, so that would be the traditional drugs. You've heard of Inflectis and Pharnex. Those companies have been in the space for a while. There's a new company called Orthogonal that we're in testing with. And there are 3 major companies, again, that are doing testing and are talking about clinical trials with us and advisors in that space. So is it working? Yes. If we had drawn this picture 10 years ago, there would have been very little on the left side and nothing on the right side. 5 years ago, we had developed a lot of the capabilities, maybe half of what we have today, and we had 3 or 4 key partners on the right side. As you can see today, It's a very rich ecosystem. And this is the one thing that gives me hope for you, for the community, for my son, is even though those things are risky, many other things are not going to go to the end, there's so much going on in so many different technologies, then it can give us real hope that basically we are on the right track here. And by the way, I didn't put everything here. I didn't put companies that work on like HDAC6 and all those nerve things that John talked about. I didn't bring, those are only the companies that are actually working with us today. And again, this picture changes all the time. In fact, I wrote this slide on Monday, and there's a couple of things I learned yesterday would actually change the slide. So this is a very dynamic picture. It's a very exciting space right now. My last point is, as we get closer to clinical trials, your role becomes bigger and bigger. I always say STAR, when I talk to the committee, is for you and by you. You guys funded this from the very first day. All the money that we put into work to make this happen came from the community. And this continues to be the case. That's why we're such an efficient company, so highly rated, because we don't spend a lot of money on fundraising. It comes from you. But more and more, and Catherine will talk to you about this, companies want to talk to you. They want you to be involved. And it becomes a really big part of how we can help them. So I will stop there, say we're excited. There's a lot of work left to do. It's not time to get the foot off the pedal, but clearly we're very excited about where we were a few years ago. Thank you.
Thank you ever so much, Gilles. I really appreciate you sharing that with us all. What we're going to do now is we're going to move to our Q&A section. If I can ask you to stop sharing your screen, Gilles, and I'll go back to sharing mine. And actually it's me next. Sorry about it. I got ahead of myself. So I just have a couple of minutes here to share a little bit of information about the CMTA's Patients as Partners in Research program, which launched in 2018. The goal of Patients as Partners in Research is to advance the work of the CMTA's Strategy to Accelerate Research, or STAR, which you've been hearing about today, and to get you, the patient community, involved in the critical work of furthering the development of treatments and ultimately a cure for CMT. We have some additional objectives underneath that main goal, which is to engage you, the CMT patient community, through supporting your access to surveys, natural history studies, and clinical trial opportunities when they begin. To inform you, to let you know around what is happening in research progress through webinars like today and through the information on our website, not just in 1A, but in other cross-type initiatives that might benefit multiple types of CMT. And we also collect and report on patient data, and that helps us to support you better. And we use this data to support and inform research, companies that we work with, and the wider CMT community. And we give you this information back again, so you can understand more about the patient community around you. So if you want to find out more about Patients as Partners in Research, this is where you go on the CMTA website. This is where all of the active participation opportunities are. There are currently 14 opportunities on the website today. And so far, we've helped to recruit CMT patient partners to 22 studies. All of this research is not possible without you. So we're really grateful to everyone who's taken part so far. So on this page, you'll find a link here to building your own profile, and that's what the profile builder looks like. So if you haven't done so already, please build a profile on Patients as Partners. It only takes a few minutes, and one of my colleagues will put the link to the Patient as Partners page and the profile builder in the chat so that you can follow that link and sign up today. We also ask that you please get your family members with CMT to build a profile too. Because 1A is inherited in a 50-50 pattern, you've probably got somebody older or younger than you in your family who has CMT. And the more we understand about each member of your family and the community, the better we can support you. So everybody needs their own profile. In return, when you join that Patients as Partners in Research community, we'll send you our monthly research newsletter and research updates that are tailored to your CMT subtype. You will be the first to hear about new natural history study and clinical trial opportunities for 1A. And you'll be invited to join any patient focus groups and webinars just like this one today. You'll also be joining over 6,200 other people who have CMT by helping to create that largest available CMT patient-reported data set in the world. And if you want to know what you could do to help CMT 1A research today, then filling in this Patients as Partners in Research profile and getting your family members to do the same is one of them. So With that, we're going to move to the question and answer section. I asked at the start of the session for you to add any questions that you had to the chat, and Gina and I are going to select a few questions to put to our speakers. We might not get to them all in the time we have, but we will do our best. And please understand that we're not able to answer any medical or personal questions or provide that medical advice. So please keep your questions general. Let's have a look and see what we have. If Laurel or Gina wants to come off mute and shout the first question out, then I can catch up on what's there.
Yes, Catherine, I've already gone back up to the top. And the first question is, this might be for John. What were the AEs reported with the Novartis medication, if this is reported in mice? I don't know that we have that information. John, are you still on?
John
John.
John wasn't able to stay for the chat because he had to go to another meeting. We don't have that data to hand. I know some people were asking about Novartis, what the AEs are, what the treatment would be like, how many doses, those types of questions. At the moment, we don't have that information, but that will be released as soon as that organization is able to move towards the clinical trial as part of their application to the FDA to run that trial.
Yes, and so that also answers the question. We do not know when that trial will start, but if you stay tuned and you're signed up to receive research updates from the CMTA, you will know as soon as we know. So that also, Madison, we do not know the dosing at this time. So Caitlin asks, for a successful trial, would muscle come back? That might be a Dr. Herman.
Yeah, David, for a successful trial, would we see muscle coming back? I mean, that's a long answer to a question, but I wonder if you could give a brief answer. You're still on mute. Oh, I don't know if David can hear me. I can answer that. David's not going to join in. He's typing. Maybe he's typing his answer.
Oh, he might be.
So each trial will have its own success criteria. So each company will be measuring a different set of functional outcome measures and endpoints. So some of them might have, achieves a faster time in a 6-minute walk test or a 10-minute walk test. So every trial will have their own outcome measure. Return of muscle, it's really going to depend on the final outcome and what the mode of action of any treatment is. And David is unmuted. Thank you, David.
Yes, I think realistically, it depends on the stage of disease. So for individuals who have milder degrees of muscle atrophy, I think it might be feasible to have some muscle come back with certain effective treatments. I think when muscles in the lower part of the leg, for example, if they've extensively replaced by fat, for example, in the muscle, then the likelihood of having significant return of muscle becomes much less there. However, there may be muscles higher up in the lower limb that are more mildly affected that could see improvements and might yet result in improvements or stabilization in things like walking and balance.
Thank you, David.
Thank you, Dr. Herman. Next question from Don, and Dr. Herman, this is probably for you as well. If the level of CMT progression is advanced, will that disqualify someone from being considered for clinical trial? Oh, did we lose Dr. Herman? Dr. Herman, can you hear me?
Yes, I keep muting myself, so I'll stay unmuted. Sorry, I missed the last bit of that question.
Oh, that's okay. If the level of CMT progression is advanced, will that disqualify someone from being considered for clinical trial or from having the medication be effective once approved?
Yes, I think that's going to be different depending on the clinical trial. So I wouldn't like to hear during this brief webinar exclude anyone from a clinical trial. But I think that some of the treatments will likely be more applicable to early administration in children, for example. Others might be applicable to administration at any time during the course of CMT1A. Generally, the FDA requires that initial treatments be tested in adults if there is an affected adult population. So I do anticipate The initial trials will be in adults for most of the therapies, but I think there'll be an effort to rapidly move trials to younger individuals when there's evidence for safety.
Thank you, Dr. Herman. Kelly, that answers your question as well. So I'm going to keep scooting down. Similarly, for adults in their mid to late 60s, when gene therapy is approved, will it be effective due to age and damaged cells? So same concept, but with gene therapy, Dr. Herman.
Yes, I mean it's hypothetical, so we'll see which treatments are effective at different stages, and it might be that there's certain treatments that are more appropriate for individuals earlier in the disease, and there may be a different set of treatments for individuals who are a bit older. But I think that I don't think we should discount the benefit that even if individuals are older or might have more advanced disease, some of these treatments may still be able to slow or stop progression. So I think the goal is really to develop treatments that can be applicable to the broadest range of individuals affected by CMT1A.
Thank you.
Just like to add to that, just because an individual might not be included or meet the criteria for inclusion in a clinical trial, it doesn't mean to say that once that drug or treatment is approved for wider use, that they would not be able to access it. So then each case would be assessed and you'd be talking to your neurologist about that. So please don't feel if you don't meet the criteria for a clinical trial, that means that eventually you would not be able to access that treatment.
Thank you. Next question from Brandon. Dr. Herman, probably for you as well. Is DTX 1252 more promising than PXT 3003? Probably hard to answer.
I think that is difficult to answer. I do anticipate, although I'm not directly involved with the PXT trial, that I would think in the next 6 months or so, I would imagine that we would anticipate learning something as to whether that a treatment, which is an oral treatment, will be effective or not. But I think that these are very different approaches, and as Jules Bouchard said, I think there'll likely be more than one strategy, and some individuals ultimately may be on some combined therapies in the future. I think it'll just be a matter of time and conducting these clinical trials really to know.
Thank you.
To add to that, we are expecting the results from Pharnex's phase 3 clinical trial in Q4 of this year. So if you fill in a patient as partners in research profile and we know you're 1A, when we get those results, we will share them with you. So that will be coming out as an email alert from the CMTA.
Thank you, Catherine. Next question, I believe this is for Nikolai. Can AAT improvement in animal testing be elaborated on? Could you talk a little bit more about that? Is there axon improvement before and after? Nikolai, are you still with it? Oh, he's unmuting. Okay.
Hang on, I'm trying to unmute him. He's talking, but he's not unmuting. There we go.
Here we go. I managed to do it.
Thank you.
So to elaborate on axon damage, yes, there is in the mouse model, there is axon damage, and we measure axon damage as the compound muscle action potential. So the CMAP, that's a typical way to measure it. So in our example, for example, it goes down to 4, and we are able to restore it to about 6, where the normal healthy level would be 8. And that goes across parameters. I also just take the opportunity to say, we did this short study in rodents because this is alpha-1 antitrypsin is a human protein, and it takes 2 weeks to see the therapeutic effect. Unfortunately, we couldn't do the study for longer. It's because the mice start developing antibodies to the human protein. And how does that relate into recovery in CMT patient? That we'll need to see from a clinical trial.
Excellent. And one more. Go ahead, Catherine.
Sorry, Laurel, it's just one other question we had for Nikolai, and it might be difficult to answer, is how long before your treatment might be offered to the public?
Yeah, again, this very depends on the how fast we can move into clinical trials, and this is definitely one of the things that we are pushing, and it depends not only on us but on the regulator. Dr. Herman is doing a very important work in preparing us for a successful clinical trial. But time will tell, but hopefully very soon.
Wonderful. And so you probably are not able at this time to speak to the side effects of AAT in humans.
Well, in fact, a lot of studies have been done on AAT, and it has no side effects. In fact, it's a naturally occurring protein, so we all have it. We all produce it in certain amounts. What we need to do in a clinical trial is increase these amounts above the normal levels that we're all producing. This is what the trial will be about. So no known side effects, which is really the beauty of this molecule.
Okay, and then another one. How do you expect the 2-week timeline for these improvements from AAT therapy in the mouse models to translate to humans? And do you have expectations and timelines for improvements in patients?
Yeah, so I mentioned that in the mouse model, the disease progression is very, very fast. So within a couple of weeks, you can already see the effects. Obviously, CMT1A is a long life disease. So it's very difficult to extrapolate, but any clinical trial design will observe a patient for at least a year, and we expect that within this year, we will see a significant improvement.
Thank you, Nikolai. I think with just owing for everybody's time and being respectful of that, I think we probably need to go to Gina Sweeney with her message for us all and to help close the session. For everybody who's asking, yes, this has been recorded, and a copy of the recording will be sent to everybody that's on the call and everybody that registered. So thank you, everybody. I'm going to hand over to Gina.
Thank you.
Gina, you're muted.
Yes, I was trying to get unmuted there. So first, thank you, Catherine, and a massive thank you to all of our guest speakers for taking the time to speak and to present to our community. And as Gilles said, that we hope this town hall gives you real hope for the future. And if you're anything like me, butterflies in your belly for generations to come. So I also want to note, I was just looking through the names. If you're on this call and you don't have 1A, know that a treatment for any type will just open the floodgates for all types of CMT. And you're more than welcome to reach out to me to get an update on your particular type. One of the things that Gilles also mentioned is it is us, our community, the patients and the families and the friends that are the driving factors in our research and all of the advances in our research. It is you are the ones that are solely funding STAR. The CMTA does not receive any grants to make this happen. It is our community. So thank you all for funding the CMTA and bringing this presentation to light. So because of today and in celebration of the 1A town hall meeting, and because there is nothing more rewarding than supporting the CMTA, Sarah Gentry, will you please pull up my slide? With the help of a 1A donor, we are kicking off a 1A $100,000 72-hour dollar for dollar match. We have 72 hours to match this $100,000. All gifts will be allocated to 1A research, and this is a perfect opportunity for you to consider giving to the CMTA, or maybe it's an ask to others to give for you, maybe an aunt or an uncle. Because remember, giving or asking for the CMTA is about being able to walk without worry of falling. It's about walking up the steps and walking back down. It's about being able to button up your shirt. It's about dismissing any fears about the road ahead. It's about knowing you and your loved ones will never have to worry about CMT again, ever. So to kick things off, you'll see my daughter Riley there. I am personally going to give $1,000 for my Riley. My question to you is, who will you give for? So go to cmtausa.org/1A match, or email me at gina@cmtausa.org for more information. With that, I thank you all for attending and for your time. And a recording of this meeting will be sent out within the next 24 hours. I hope you all have a fantastic rest of the day. And again, thank you. Take care.